Welcome, everybody, to the Scientific Roundtable. We have a pretty full schedule with four speakers today. The agenda for today is basically focused on abstracts that were submitted to the symposium, and these will be junior people presenting updates on research that they're doing. So the talks are set to be for 10 minutes with 5 minutes of questions. So please, at the end of the talks, unmute yourself if you have a question and open a dialogue with the speaker directly. We will have to limit you to that amount of time because we have four speakers and about 15 minutes to get through each one of those. So please be aware of the time. So with that, I'm going to start it off with Tori Flournoy from University of Alabama at Birmingham, who is going to be telling us about democratizing cystic organoid models for pathway analysis and compound assessment. So Tori,
Thank
she's
you.
on.
Pre-order.
Yep, there you are.
How you doing?
Perfect. Looks good.
Great. Okay, thank you for having me today. So I will be talking about my current project, which is democratizing cystic organoid model for pathway analysis and compound assessment. So I want to give a background of what is ADPKD. Most of us already know, but autosomal dominant polycystic kidney disease is among one of the world leading genetic kidney disorders, generally characterized by renal fluid cysts. And as you can see in the bottom left, a polycystic kidney is nearly double the size of a regular kidney. And as you can see on the bottom right, the large renal fluid cyst in the actual kidney leads to end-stage renal failure. So this is a very high demand study in this disease, as only one FDA-approved drug is out there, and it's tolvaptan, which has a number of side effects, which has a high demand and cause for research for it. And so I want to give a background is what is the organoid and why do we use that as a disease model? So the use of HPSCs, human pluripotent stem cells, we usually use these and generate mutations in the PKD1 or PKD2 genes. And we use these organoids as a disease model. It's a 3D disease model that allows us to culture these organoids and generate thousands of organoids to be able to study the ADPKD and polycystin 1 and polycystin 2 as using humans and mouse can limit this process. So we start with CHR, which is a GS3 inhibitor, which allows for the Wntz pathway and beta-catenin to be activated and allows for the late primitive streak, and then further activin A, another growth factor, allows for the formation of the intermediate mesoderm, and then also continuing to the metanephric mesenchyme, and then continuing using FGA and CHR. Now you have the continue of these renal tubules and renal vesicles, and then the formation of nephrogenesis. So now we are getting to the process of actually making a true kidney with podocytes. And this protocol has been optimized from previous organoid developers such as Dr. Freeman. And so now I want to talk about how we democratize these organoids. So we want to be able to generate wild type and PKD 1 or 2 mutants and be able to send these organoids out for actual, screening and pathway analysis for other labs. And we are able to upscale organoid production in our lab by generating thousands of organoids per plate using 12-well easy spirit plates. And so this is just a schematic of how we will freeze, send, and you have a carrier choice to get to your lab. And this will allow for the process of skipping organoid development, and you only have to thaw them when they come ready. So this is just a process of cryopreservation of the organoid, which I optimized from Dr. Yuri Mirozani. Using this protocol, I use equilibrium solution, which allows me to freeze get the organoids prepared for freezing, and we use a rapid vitrification solution. So it's a rapid freezing solution, which allows us to submerge in liquid nitrogen for 30 seconds without damaging the podocytes or tubules in the organoids, to allow them to be shipped across the nation to a desired lab they need to. And all you need to do is thaw these cells with a specific thawing solution, which is sucrose, and this allows the organoids to come back to life. And we can plate these in 6-well, 12 wells, and 96-well plates for further compound testing and tracking. So I just wanted to show a PKD GFP cystic outgrowth over one week. We wanted to determine how well our cryopreservation is working. Are these organoids surviving? And as you can see, we have a MAFB EGFP podocyte marker to show you that our podocytes are able to survive. And we also have cystic growth from a PKD1 organoid. Currently, we are working on freezing the wild type organoids to determine how well they freeze and if they do form cysts. And so we also want to democratize the cysts from these organoids, since generating an organoid is very long in cost and reagent consuming. So skipping that process and actually being able to thaw out cysts and grow cysts from your own lab without going through that whole process. So we dissociate, we freeze, and you plate the cystic structures. And these cystic structures can be shipped to many labs around the nation, so they can be thawed upon arrival and cultured. So here's a process of just how we regrow and passage these cysts. So what I would start with is delayed and detached cysts, and then we culture these in 6-fold plates, and then when they get to a centimeter in diameter, we let them grow more, and then further, I make a dissociation buffer. We dissociate these cysts from the original organoids, and we dissociate them into single cells, and then we can freeze them. And then after we freeze them, we can thaw these cells and then culture them in 12 wells, 6 wells, and 96 wells as well. And so we do a cell count. So we have typically 1 million cells per vial. And you also can skip the late and detached cysts, as they allow for culture to happen easily, and you don't have to worry about these early stages. And so here's just a schematic of how these actual regrowth cysts look in a 96-well plate. So we have 3 different concentrations, 1,000, 2,000, and 3,000. As you can see, each concentration allows for robust cysts to grow, and this is over a course of 7 days. And this shows that, we are able to grow cysts from these cystic structures. And here's just another thing, that you can also change media with these regrowth cysts, optimizing our protocol, allowing you to use different compounds for compound assessment and studying which different, PKIs or different drugs can limit cyst progression and actually cyst growth, which is a good thing. And we can also grow in 6-well plates. So here's the day 2 post-thaw. You can see a large cluster of epithelial cells, and they're all over the place. By day 7, you can see a nice balloon cystic structure, and this is a nice promising view to show that the cysts are continuing to grow in the 6-well as well. And so we want to determine, okay, are we able to quantify cystic growth? Are these cysts actually growing over time? So we have this program where we use AI to actually quantify cystic growth. And so I want to determine how well the AI can quantify it. So we have a PKD1 knockout cyst at the bottom, cultured in a 96-well plate. And I compared 40 cysts from the AI and me manually counting the cystic area. And as you can see, a nice comparative normalized area growth, comparing the AI and the manual. And we use this AI system that allows us to quantify it, which is good. And I want to determine, can it do it for a large number? And so here we wanted to compare the PKD1 to the regrowth cyst. And as you can see on the right, we have a nice graph showing the actual normalized area, the continued growth of both PKD1 and regrowth cysts. So this is letting us know, okay, the actual regrowth cysts are continuing to grow comparative to their predecessors, original cysts. And so from here, I wanted to ask, how does the proliferation rate of the regrowth cyst compare to the original cyst? So how are these cells proliferating? Is the proliferation rate, are they even proliferating at all? So here we have a PKD1 knockout cyst, PKD2 knockout, and a PKD1 regrowth cyst. I am currently working on getting the PKD2 regrowth cyst stained, and that data is being currently worked on. But as you can see, a nice spread across a nice EDU comparative of each of the PKD1, the PKD2, and the regrow. As you can see to this right, you can see a nice normalized mean around 23% among most of the regrowth cysts. And I typically use cysts around the same size, which is 1 millimeter in diameter. So this allows for to eliminate the bias and to try to keep it uniform. And as you can see, the proliferation rate is very similar. So this
2 minutes remaining.
this is a good step moving forward. And so from here, we can conclude that the organoid GFP marker is preserved after cryopreservation, allowing for large-scale freezing and shipping. The cyst can be dissociated in the single cell and can regrow back as prominent cystic structures, as I showed. And the regrowth cyst in the Easy Spear 12 well plates, which laser engineered wells, allows for upscale growth around 48,000 cystic structures per plate, allowing for a large scale growth. And the proliferation rate of the PKD1 knockout and PKD1 regrowth cysts are relatively similar. And this allows me to set up my future direction, which is to do a FACS sorting with the ECAM antibody, which is the epithelial cell antibody, and do a single cell plating of these ECAM to determine, do these purely epithelial cystic cells grow from single cells? And then we can dive into different other questions from answering this question with the single cells. So from there, I also want to do a bulk RNA sequencing to determine what is the average gene expression of these regrowth cysts compared to their original cysts, and also for PKD2 and as well. And this will allow me to set up compound assessment. So we can use these regrowth cysts to test a number of compounds to see what is limiting cyst growth and possibly initiation as well. And then we can just study these regrowth cysts to study what pathways are being upregulated or downregulated in these regrowth cysts and PKD1 cysts as well. And so from here, I just want to acknowledge my LabGap song, train, my funding sources, which is the PKD RRC, the CubPrime, which is an internship I'm funded by. And I want to give a thanks to the PKD RRC Roundtable for having me today.
Very wonderful. It's now open for discussion, and I'm going to ask the first question, and this is kind of off target from what you were actually presenting on. But once you dissociate the cyst from the organoid proper, does that, if you were to culture that organoid proper, the one, so not the cystic part of it, do cysts reform in that organoid?
I have not tried that yet, and that is something that is a good question, and we should definitely try to determine, if we dissociate the organoids and regrow, will the cyst grow from the organoid? That I do not know the answer to that, though.
Robbie.
Hey, Tori, that was a fantastic talk. It was really nice to see the progress of your work coming along. I thought that it was really cool that you were looking at EDU, because I'm coming from an in vivo background. I'm always thinking about proliferation with cysts. I was curious if y'all have looked at the EDU rates in the organoids themselves versus the cysts and how they might compare.
I honestly have not looked at the EDU rate in the actual organoids, but I'm sure my PI Jack has seen it before. I would just have to see that. And I also am thinking about using an EDU on the actual organoids to determine those proliferation rates after the cryopreservation as well. So that's a good question. I need to get that done.
Other questions?
Yeah, can I have a quick question?
Yeah, go ahead.
Yeah. So as I understood it, you send out a dissociated cell from psyllium. So this is each individual separated cell. And
I'm sorry.
Right. So the cell you're going to send out to other investigator, these are dissociated, but in individual cell. It's not not a cyst. This is just different cell, right?
Yes, sir. These are cystic cells. They're dissociated all the way into cystic cells. Typically, most of them are single cells. Sometimes they form some cystic structures, but typically most of them are single cell, which is why we're trying to tease out which exact populations of these cells specifically contribute to the cyst growth.
Right. Okay. So when you regrow those cysts from those individual cells, so you have multiple cysts coming from different cells. So when you actually measure proliferation, are they similar, or do you see differences between different cells?
Yeah, that's a good question. So the proliferation rate, I usually try to eliminate bias. So I try to pick cysts that are very robust. So cysts that are actually forming and growing. So the ones that I take from the 96 well plate that are constantly growing, I take those and then I do micro dissections. And typically the EDU rate is very similar among all these regrowth cysts compared to the PKD1. I do not know if they're differences, if they're coming from like different tubules or different places, but typically the EDU is pretty similar.
Okay, thanks.
There's a question in the chat from Martin Prince. Is there a maximum size? Do some of them pop or fall apart when they get too large?
No, the cysts right now, they seem like they constantly grow. Like I start from the cystic cell, and right now I'm still culturing cysts that have been cultured for months, and they get to like 1 to 2 centimeters in diameter and even further. I haven't seen any pop from getting too big yet, but they keep growing.
But they don't ever fall apart. They remain intact and
Yes, yes,
yeah.
sir.
Okay. Other questions?
Maybe very also maybe quickly, but this is a PKD cell. Do you also thinking to send out the wild type cell, maybe from
Yes. So yes, that is the thing. But honestly, the wild types don't typically form cysts, what we per se. So we're actually studying to see if they do form cysts in the organoid, but to dissociate cysts from the organoid, I don't think that the organoid can form
1
cysts.
minute remaining.
But we are trying to send out the wild type organoids to be studied.
And when could you send out? Are there any time plans?
I'm sorry, can you repeat that?
When?
I have a number of batches prepared now, but I mean, I would have to talk to my PI as well to determine what's a good time.
So I think one of the things you guys are trying to do is to see how people outside of the organoid field can use these, right? So shipping them out, I think, would be a really important thing to do.
Yeah, yes, definitely.
All right, Tori, thank you very much for a wonderful talk. We're going to move on to the next speaker, which is Sun-Hee Huang from UT Southwestern. We'll be talking about adenyl cyclase targeting to the cilia in embryonic and adult onset ADPKD.
Good afternoon. Can you hear me?
Yep, well, your screen was there. I don't know where it went. So
Okay.
I still do not see your screen.
Okay, good afternoon. My name is Sun-Hee Huang.
Your screen isn't shared. I think there was some quick sharing, unsharing, and Tori's
Okay.
flashed and yours went away.
If you can share your screen again.
Can you see clearly now?
Yep.
Yes, thank
We're
you.
good.
Okay, thank you. Sorry about it. Good afternoon. My name is Sun-Hee Huang, senior research scientist in Psychiatric Lab, UT Southwest Medical Center. Thank you for giving me this opportunity to present my data. Appreciate. Today, I will talk about adenyl cyclase targeting to cilia in embryonic and adult onset ADPKD. The Somno's lab showed loss of PC1 or PC2 causes big cyst in kidney. Loss of cilia also develops cyst but small, and loss of cilia in the PKD loss, the cyst growth was only partially suppressed, suggesting that cilia has positive and negative complex roles in cystogenesis. What are the ciliary regulator of cystogenesis? These are published from our lab and Carol Liam's lab in Yale. TRP-like protein TRP3 using conditional knockout TRP3 mouse. Our lab used the HOXB7 CRE to see embryonic onset, and Carol's lab used the doxycycline-inducible PEX8 RTTA TETO CRE to see adult onset. In embryonic TRP3 conditional knockout, cyst was slowly developed, but it cannot suppress the cyst growth in PKD1 double knockout. Unlikely this embryonic, in the adult onset, loss of TRP3 can almost completely suppress the cyst growth upon PC1 loss. Clearly, TRP3 has a role in cystogenesis, and it is different in embryonic and adult PKD. So, cilia-dependent cyst activation CDCA. In wild-type cells, CDCA was suppressed by polycystins and is activated through unknown mechanism. In PKD1 CK conditional knockout, CDCA is de-repressed and constitutively active, signaling leading to severe cystogenesis. And TRP3 can recruit something into the cilia, which can control the cystogenesis. This ciliary localized CDCA may not cause cysts, and concomitant polycystin loss reduce the cyst. What can be this unknown CDCA? So Nephros lab showed adenylyl cyclase AC, AC5 and 6 mainly expressed in normal nephron and ADPKD. Igarashi lab and Cohen's labs showed loss of AC5 or 6 reduces the cyst growth in mouse PKD. And Welles's lab showed that AC3 expressed in ADPKD kidney cysts. We overexpressed the GFP tagged AC3 in 3T3L1 cells. These AC3s localized the primary cilia really nicely. But when we knock out TRP3, this ACs express the trafficking to the cilia was demolished. So these ACs can be the candidate for the promoting ciliogenesis. So we performed the tandem affinity purification followed by mass spectrometry. Stably expressed the lab-tagged AC3 or AC6 in 3T3 NIH 3T3 cells. In both cases, ANC-MI2 was identified combined with the AC3 or 6. And lab-tagged ANC2, the mass spec result, AC6 was showed identified. And immunoblotting upon mass spec lab-tagged AC3, 5 and 6, the endogenous ANC-MI2 was showing up, and it is showing ANC-MI2 is a common interacting factor for these all ACs. So what is ANC-MI2? Ankyrin lipid domain and zinc mint domain containing protein. It is a predominantly cytosolic protein. So what kind of mechanism is happening between ANC and these ACs? So from this slide, it is the unpublished data. So we first used the IMC3 cells, and in these cells, we overexpressed the GFP tagged AC5 or AC6. These are nicely co-localized in the primary cilia. And when we knock out the ANC-MI2, these trafficking to the cilia was significantly reduced, both AC5 and AC6. So lack of ANC-MI2 prevents ciliary localization of AC5 and 6. So the question is, if we knock out ANC-MI2 in cystic kidney, what can happen? Maybe we can see we can test a role of multiple ACs at a time. So we have ANC-MI2 conditional knockout and close with the PKD1 conditional knockout. For this study, we generated two different models, early onset, embryonic onset, and late adult onset. For early embryonic onset, we used the FOXP7 Cree mouse, and late adult onset, we used the PEX8 RTTA, doxycycline-induced PEX8 RTTA, and TETO Cree. We IP'd the doxycycline at P28, daily 3 times, and euthanized at 5 months. So in embryonic onset, at postnatal day 3, PKD1 knockout already showing big cyst, but ANC, from now on, I will call ANC-MI2 as ANC. ANC knockout exactly same as wild type. The double knockout reduced the cystogenesis. In kidney body weight ratio and cystic index, both in double mutant significantly reduced the cystogenesis. And next, we checked the survival curve. PKD1 mutant started dying at P10, and the double knockout from P15, and the value was significantly improved. Next, we checked the adult onset and HE staining, kidney body weight ratio, and cystic index. All of them showing double mutant significantly reduced the cystogenesis. Next, we did the immunofluorescence to these kidney sections with LTL positive proximal tubule, the green boxes, and aquaporin 2 positive collecting dots in pink boxes. We calculated the cystic area, both the proximal tubule and the collecting dots, significantly reduced the cystic area. And next, we checked the BUN, and total cyclic AMP levels. Both values were significantly.
2 minutes remaining.
Thank you. And next, we stained the kidney sections with Ki67 to check the proliferation. Lots of Ki67 positive red proliferating cells in PKD1 knockout, but it was suppressed in DKO, both the proximal tubule and collecting dots. Next, we stained co-immunostained kidney sections to see the ciliary length with ciliary marker acetylated tubulin. The quantification shows ciliary length in both proximal tubule and collecting dots in PKD knockout are longer than controls, and the length was significantly reduced in double KO. So the conclusion, ACs are directly localized serially localized activator of ADPKD. Lack of NQMI2 reduces serially trafficking of ACs in kidney epithelial cells, and loss of NQMI2 in PKD1 knockout mouse suppresses cystogenesis, proliferations, and ciliary length. The summary, in wild-type NQMI2 is helping maturation the ACs, and this stabilized ACs can localize to the primary cilium. And we know from Watson's labs, the photo-activated ACs inside of the cilia, increase the cilia length. So cyclic AMP levels in cilia determine the cilia length. We see that cilia length are increased in adult PKD1 knockout cystic condition. These conclude that these could be from high cyclic AMP, from total cyclic AMP levels, or from increased ciliary levels starting from pre-cystic conditions. And in DKO, the ciliary lengths are not increased, suggesting that low cyclic AMP in cilia contributes to this effect. These results suggest a pre-cystic ciliary pathway contributing to cystogenesis. Thank you to my PI, PsyCat, and lab members, Kyung-Suk, Yu, Haymond, Vivek, Kevin, and also collaborator Professor Kwan in Maryland School of Medicine, and Patricia in PKD RRC. Thank you for your attention, and please give me any question and advice. Thank you.
Thank you, Sunny. Open for discussion. Any questions? I'm going to start out with a question on the cilia length issues. Do you think that changes in cilia length are a cause of the disease pathology, or is it just a consequence of this altered cyclic AMP within the?
That is really.
Yeah, the chicken and egg and cause and consequence.
Answer the question. Yes. So we know the PKD patients in kidney or mouse kidney, PKD kidney has a really high cyclic AMP levels. So we checked it in double KO. These ACs are not functioning. If the ACs are not functioning, they cannot produce the cyclic AMP. So main thing is, if we can reduce the cyclic AMP or ACs activity, we can reduce the disease.
Jeremy has what I think is a question in the chat. The ANK2 single knockout kidneys are pretty normal with a question mark. So I think he's asking, is there a phenotype in the ANK2 kidneys?
So it is a different, that is a really nice question. And embryonic, at P3, it is significant, but after I can show you the result. At P15, the looks like the
I think he's asking specifically in the ANK2 knockouts.
Oh,
right? Is there
ANK2
any-
knockout. No. The ANK2 knockout, sorry. ANK2 knockout doesn't have any phenotypes.
No phenotypes at all.
No, it looks like exactly like wild type.
Okay. It's interesting. Other questions? Too quiet.
I am trying to publish, submit this paper, these data to journal. So if you have any recommendation or question, please email me or sending me email or like that.
All right, if there's no other questions, Sunhee, thank you very much for a enlightening talk, and I'm going to turn it over to Terry for the remaining two.
Okay, hello, everybody. Our next speaker is Sarah Miller. She's from the University of Oklahoma's Health Sciences Center, and the title of her talk is A Cross-Model Spatial and Single-Cell Atlas Reveals the Involvement of SPP1 in the Progression of Polycystic Kidney Disease. Thank you, Sarah.
Hi, thank you, Dr. Watnick. I tried to turn my camera on. It doesn't appear to be working, so I apologize for that. And also, I have had some internet problems. I'm actually at a conference in Virginia, so I also apologize if things freeze up a bit. As Dr. Watnick said, I will be talking about my project looking at SPP1 in polycystic kidney disease. I am currently a postdoc in Kurt Zimmerman's lab. Actually, will be transitioning to faculty soon, however. All right, so I'm just going to give very brief background. We all know PKD. It's a multisystemic disorder with many different etiologies. It affects approximately 1 in 500 people, and it leads to cysts in both the liver and the kidney, eventually causing end-stage kidney disease and liver fibrosis. Now, unfortunately for people suffering from this disease, there are very limited treatment options available, including tolvaptan, or one FDA-approved drug, renal replacement therapy, and then transplant. Something I really want to emphasize here is that there's many different factors influencing the rate of disease progression. So we know that the age and sex of the patient, the identity of their specific gene mutation, and then many environmental factors can also impact the disease progression. And there's just a lot of heterogeneity present in the patient population. So patients are stratified into various risk classes of disease according to how rapidly their disease progresses. And we know that patients, even some patients with the same gene mutation, can have dramatically different presentations of disease. So in order to try to kind of encapsulate this heterogeneity that was present in the patient population, we sought to establish a single-cell sequencing atlas comprised of many different mouse models of disease. So in our atlas, we use 6 different mouse models of disease, and these included a variety of models, as you can see here shown on the slide on the left. We have things like the PAX8 model all the way to the IFTD8 or the PKHD1 CRE model. Now, the y-axis in this figure is giving you the age of the mouse in weeks, and then the arrows that are present indicate when the disease starts, as well as when the disease ends in these respective models, with the length of the arrow roughly representing the length of the disease that they suffer. So again, we have models that are orthologous to ADPKD patients as well as non-orthologous, like our IFTD8 model. We have mouse models that are slowly progressing, representing those lower classes of our ADPKD patients. And then we also have rapidly progressing models in this atlas. And then we also have models that have a global gene deletion as well as epithelial cell specific gene deletion. So again, what we're trying to do here is really represent the heterogeneity that's present in our patient population by kind of forcing all of these different mouse models together so that we can ask, is there a similarity? Are there signaling pathway similarities that are present across these PKD mouse models? So when we combined all of these models together, we end up with 41 samples. We have 19 control and 22 PKD, and we had a total of 265,000 cells. These represent, all of these cells represent both the stromal compartment as well as the immune cell compartment. Now, in this project, we're specifically interested in the stromal cells, in particular, the tubular epithelium. So when we subcluster our tubular epithelium, we see we get 12 different clusters, and these represent all of the different segments of the nephron. So we have everything from our S1 all the way up to our intercalated cells, nephron connecting tubule, good representation across our nephron. And then when we looked at the frequency abundance of these clusters in our control samples versus PKD, we found, interestingly, we see a couple of these clusters that are more abundant in PKD. So we see things like our lower limb loop of Henle, our thin descending limb loop of Henle, principal cells, and then our S1 through S3 segments were found in a much higher frequency in those PKD samples when compared to our control samples. So suggesting that these clusters might be impacted in PKD. Now, in order to more closely interrogate the differences that are present across clusters among our samples, we looked at the top differentially, or the number of differentially expressed genes across clusters. And using both p-value and adjusted p-value, we see 3 clusters really stand out with the number of differentially expressed genes in PKD samples. These are our principal cells, our lower limb loop of Henle, and then thin descending limb loop of Henle. So again, these are 3 of those clusters that were also more abundant in our PKD samples. Looking at these 3 clusters, in particular, we wanted to look at their top differentially expressed genes. So in these heat maps, I'm showing you the blue squares and the columns beneath them are representing our control samples, whereas the orange squares and the columns beneath them represent our PKD samples. So looking at these 3 clusters, we can see our top differentially expressed genes show things like HAVCR1 and lipocalin-2, so kidney injury associated genes. We're seeing immune cell related genes like CSF1 and complement like C4b. And then we're also seeing that gene identified by Yoshimoto and Ben Humphrey's lab, GPRC5A. So again, these genes are all kind of associated with PKD, so strongly supporting that these 3 clusters might be involved in PKD. Now, one of the next questions we wanted to ask is what variables are driving these differentially expressed genes in our nephron segments that we're seeing? So we plotted these differences in our control and PKD samples as a function of either the sex of the animal that we're looking at, the model, or whether the model was orthologous or non-orthologous. And you can see we have some differences, but there's really not dramatic differences when we're looking across PKD samples and then comparing those to our control samples. As in, when we look at this, we see kind of mostly orange in the PKD and mostly blue in our control. So really not dramatic differences present. However, when we looked at these differences as a function of cyst status or cyst rate, so by cyst status, I mean our PKD cystic samples were animals that were already showing cysts. They were cystic at the time of harvest, whereas our PKD non-cystic animals were animals that did not have cysts, so they were non-cystic, even if they might be lacking those cilia-related genes. And then the rate of progression just applies to how rapidly their disease is progressing. So these were really the top 2 factors that seem to be driving these differentially expressed genes in our nephron segments. Now, one of the next things we wanted to look at is our overall signaling pathways that were different across these various clusters identified in our tubular epithelium. And interestingly enough, we find that osteopontin was one of the top signaling pathways identified across clusters. So this included not only those 3 clusters that I mentioned earlier that really stood out to us, but also a nephron connecting tubule. And it just seemed to be upregulated across all of the clusters actually in our PKD samples. We next wanted to know what cell types were involved in this SPP1 signaling. So we looked at our SPP1 signaling networks in PKD samples and control samples from these clusters, and then to see what cell types they were signaling to. And we find that in our PKD samples, SPP1 signaling seems to be primarily communicating with fibroblasts and then various immune cells such as Ly6CI monocytes, CDCs, and then our early EMT stage fibroblast as well. So these are 2 cell types, both the monocytes and just immune cells in general, and then fibroblast. We know that these cells are implicated in PKD disease and progression. So this, again, supporting that SPP1 might be involved in our cystic disease. So the next question we had with this project was then, what is the function of SPP1 in cystic disease? So what's it really doing? In order to address this, we use 2 different mouse models, our IFT88 mouse model and our PKD RRC mouse model, and cross them to an SPP1 knockout mouse. And interestingly enough, when we crossed the IFT88 mouse model to these SPP1 knockouts, we saw an increase in kidney weight, body weight, cyst index, and cyst number. However, in our PKD RRC mouse model, we actually saw a decrease in kidney weight, body weight, cyst index, and cyst number. Looking at fibrosis and then BUN, we saw no change in our IFT88 SPP1 knockouts, and in our PKD1 RC animals, we saw a decrease in BUN, so mimicking the results that we just saw. So it appears that SPP1 knockout in IFT88 makes disease worse, and in PKD RC, it makes disease better. So what is happening here? Why are we getting two differential effects? Well, to answer this, we went back to our single cell sequencing data and looked at the expression in our tubular epithelium and non-tubular epithelium. And what we see is that in the IFT88 models, our tubular epithelium signaling doesn't seem to be changing that much in our IFT88s, whereas in our PKD-RCs, it does look like we're getting both an increase in signaling intensity as well as an increase in the cells, the percentage of cells expressing SPP1. When we look at the non-tubular epithelium, however, the pattern's a little bit switched, and we see more of an impact in our IFT88 mice than we do in our PKD-RC mice, suggesting that our SPP1 expression might be coming from different cell types in these two disease models. And lastly, we wanted to look at what cell types are being signaled to with SPP1 in these two mouse models. So in our PKD-RC mice, we look in our tubular epitheliums, and that's where we're seeing the effect. And we see that it seems to be signaling with kidney-resident macrophages and Ly6C high monocytes. When we look at our IFT88 in the non-tubular epithelium, we see signaling instead to fibroblast and Ly6C high or Ly6C low monocytes. So it really seems like these disparate outcomes could be due to different cell types. Receiving the ligand. And there's my summary. Thank you guys. Sorry, ran a little over. I'll leave it on the summary slide so y'all can read it one more time. And I will take any questions.
Okay, open for questions.
Go back to my acknowledgements as well.
Okay, thank you. Anyone? Okay, I guess I will ask oh, okay. Oleg, go ahead.
So I have one small tricky question. So that's it's not directly to apply incomplete in this story. It's nice that you they use a lot of effort and do all this transcriptomics and on a single cell level with many models. But just to put some awareness about that, actually, how many cells was pick up? And it looks like you have like 250 cells, but how many nephrons is that in a calculation? Recalculate to nephron amount. How many nephrons it will be translated for all the animals what you have? How many? Just 40 animals you have in the groups, and how many nephrons actually you count in your transcriptomic analysis?
So you're asking how, like, what the sequencing depth is essentially? I mean,
no. If you come back to your slide quickly.
Yeah, 200 with 260,000 cells.
Yeah.
I mean, I don't think there's a way to know. I mean, we have on average 1 million functional units per kidney. So
this
clearly,
is I'm just saying that we should always remember that doesn't matter.
yes.
You analyze approximately less than 1%.
Yes. Oh, yes, exactly. That's a very good point to
Close
make.
to 0.1% of the cells.
Yeah.
And it's like a prediction. It's a great technique, but when you get this SPP1 gene pick up in your analysis, you need to plan the next step to see functional to explain this. Yeah, probably.
Yes.
Other functional analysis to get knowledge more deeply what this actually means.
No, yeah, I
just my suggestion.
Yeah, it's a very good point. It's something we all need to remember. I mean, even the way we prepare our samples can impact, what cells we're getting on the other side. So it's something to keep in mind.
Any other questions?
Sarah, I have a quick question. When you looked at the differential expressed genes, I noticed things like the distal tubule. There was no differential expressed genes from the distal tubule. Yet I think that's an area where we do get cysts in a lot of these animals. Do you have any ideas on why?
No, I mean, not particularly. So
I mean, it just stood out to me that you actually had like 0 differential expressed genes from the distal tube, which
yeah, so I mean, I would maybe hazard a guess because looking at all of these mouse models combined, that might be why. If you look at each individual mouse model, when you compare the PKD and the control samples, you'd probably see differences, maybe
but
more.
there was no correlation with, say, where the cysts actually start to form in the different models. Like I think the juvenile ones are a little bit different than the adult ones. And stuff.
They are, yeah. No, they definitely are. And because what we were looking at with this atlas in particular is similarities across models, those differences aren't necessarily going to pop out. Like even if they're present in the juvenile versus the adult, we're looking at similarities across all of them. So those aren't going to be, you know, what come out to us.
Thank you.
There is one question in the
1 minute.
in the chat. Basically, interesting to see, this is from Yoshi, interesting to see enrichment of the thin limb of Henle loop and PKD. Did you see cystogenesis in the, yeah, I guess, did you see cystogenesis in the DTL, in the distal tubule? I guess that's related, Brad, to your question.
Yeah, where are we seeing the cysts pop out? So in the 2 models that we have done, they seem to be primarily like cortical, but so I'm not sure about the other animal models that are present in the atlas, because a lot of these were publicly available data sets. The PKD RRC and the IFTD8 models are the ones that we've done in lab. So
Okay.
I'm not sure about whether they're in the loop of Henle and the other models.
Okay, so let's move on. Thank you, Sarah. That was a great talk, and we'll move on to our next and last presentation. Okay, and the next presentation is Ching Shang from Weill Cornell, and the title of the talk is Trace.org, an AI platform for reproducible kidney, liver, and cyst contouring on MRI scans from ADPKD patients.
Yeah.
So with that.
Hi everyone, thanks for being here. My name is Qing. I'm a computer scientist, so my talk will more based on the technical aspects. So I'm here to introduce Trace, an AI platform for reproducible kidney, liver, and cyst contouring on MRI scans for ADPKD patients. As you all know, TKV is a critical biomarker for ADPKD disease tracking. It's also used to calculate the Mayo image classification, liver volume, and cyst growth to determine how fast the cysts are growing. However, these measurements are hard to obtain with intensive human efforts. So we did a survey over 145 CT and MRI reports from ADPKD patients. What we found are interesting. Only a quarter of the MRI reports included kidney volume. None of them included Mayo classification, and 1% of them mentioned liver volume. We found radiology reports are often missing total kidney volume. Even when the kidney volumes are reported, we know from the literature that the calculation using the ellipsoid formula with kidney length, width, and height isn't so reliable. It leads to an inter-reader variability of plus or minus 15%. Manual contouring is better. It brings down the variability to plus or minus 6.7%. But that still is not good enough for ADPKD patients where the kidney growth rate is only 3 to 5% per year. So why there are so many variability in volume measurements? We can just show you an example here from the same patient where the right kidney is painted in red and left kidney is painted in green. In the axial scan, the volume measures 1565 cc, while in the coronal scan, just a few minutes later, the volume reads differently with 1750 cc. That's a 10% difference, even though the 2 scans are done on the same patient for the same visit. And this has to do with various other details about the acquisition. For example, the MRI optimized image for looking beautiful, but it's not optimal for volume measurements. The image is beautiful on the coronal plane, but when you reformat to a different plane, you can see the resolution is poor. And it's also hard for patients to hold the breath for the entire abdomen and pelvis. And if you look carefully here, you can see the glitches. That means this was actually a series of multiple breath holds, so they don't exactly line up. Minor motion during the exam can also modulate the image intensity and create a measurement error. The overlapping slices has the wrong thickness here that also artifactually increase organ volume due to the slice thickness. So what we can do to reduce such variability, we take all available sequence and average them. That will bring down the variability by the square root of the number of MRI sequence being averaged. In this case, is square root of 5, which is 2.2-fold. And this wouldn't be feasible manually, but with our deep learning model, it's easy and fast. Using this, we can see a 1.3% test-retest reproducibility. We evaluate our model performance on 20 MRIs for kidney, liver, and compare that to 2 other publicly available segmentation models, total segmentator and auto MR annotator. And our model is outperforming the others with a DICE score of 0.98 for kidney and liver. And here is an example where the tracework is correctly labeling the kidneys, while on the other side, you can see that total segmentator and MR annotator are not recognizing the polycystic kidneys. It makes sense that we are trained on over 500 ADPKD patients, whereas these other models are probably trained on normal cases. So validation was also performed on external data from Bergamo, Italy. We can see the results are also promising for kidney and liver with a DICE score of 0.88 to 0.95 and 0.83 for the kidney cysts. And for the liver cysts, the DICE score is better on the larger liver cysts. In addition, special thanks to Dr. Anna Connolly, who did further validation on a data set from 6 centers on 73 MRIs, and they achieve a similar result. So the model code is available for usage. You can scan the QR code or use this link to access it, but we know it's hard to use if you don't have any coding experience. So we built a tracework, a website where you can just upload your image in DICOM format and get the volume report automatically. All you need to do is to sign up for an account and sign in. We have a 2-step verification for sign in, and everything is encrypted on the website for security. So once you're in, you can upload your DICOM image, then the website will strip away all the PHI in your web browser before anything is uploaded to our server. So it's HIPAA compliant. So please remember to put any pseudonym for remembering this patient. If you want the Mayo classification plots, just include the patient age and height, which are essential for the calculation, and hit the submit button and give it some time because processing will take a couple of hours depending on GPU availability and the size of your image. Once the results are ready, you can just click the view PDF for viewing the report. Here is an example of the report. Here is the pseudo patient name that you just entered, and it will include some snapshots of the organ segmentation, cyst segmentation, and a summary for the kidney liver volumes and also the cyst volumes, and also the Mayo classification plot. You can also download the zip and extract it, which will give you the segmentation mask along with the report PDF. Please be aware that we are not FDA approved, so this is a tool designed for research purpose, and the results should be reviewed and refined for correction. For viewing and refining, we recommend using the ITK Snap, which is an open source tool. And here is some
2 minutes remaining.
if you want to view more. Let me provide some preview of how of the future work we are trying to include. We're looking to include all other organs, including IVC, stomach, and for cysts. We are trying to separate the complex renal cysts, simple renal cysts, and include pancreatic cysts, etc. And then we also label the fat and muscle index labels to try to calculate the visceral fat and skeletal muscle index. Another feature we are developing is tracking the volumes across different time points so we can evaluate whether the tolvaptan is working. For example, in this patient, the growth rate is 3.4% prior to tolvaptan. After the treatment, it was down to 1.1%. Here is another example that the tolvaptan is working. And here is an example that the tolvaptan has no benefit on this patient. We just published this paper in JCM recently. If you are interested in that, definitely check it out. Yeah, thanks. And open to any questions.
Okay, questions? Well, I'll start. Nice presentation. So right now, this is primarily for research, right? Is that okay?
That's
And have you, I mean, it seems a little complicated. Have you had anyone use it yet, like from outside of your institution?
yeah, we have over 10 users right now there, and they uploaded probably around 200 cases already. So it's a great validation for our website as well. And they are from various countries as well. Some of them are from Italy, some of them are from Ireland, and also Australia, and also from America, like other states.
Okay. Any other? Oh, I have one more question. For the exophytic cysts, which can artificially increase the total kidney volume, how do you deal with that? How does the program deal with that, or is that in the future?
Yeah, we are including that as a part of our segmentation results, but since it's not validated to be perfect right now, so we're trying to make it perfect and release that to our website as well. The labels, yeah.
Any other questions out there?
Harry, there's
Oh.
one in the chat.
Oh, there it is. Okay, I see it. Great talk. For the input of your model, is there resolution limitation of the image?
No, there's no resolution limitation, but because we are trained on different resolution data, training data, so we can handle even low resolution data. But it is recommended to use higher resolution because that will definitely give us a better results.
Any additional questions?
I have one real final question maybe, but so looking at temporal analysis, is there a way of measuring individual cyst growth or retraction rates, or is it just volume?
Yeah, that's actually a good question, because we are trying to separate the cysts into clusters, trying to understand and predict how the number of cysts in these patients. But that is really challenging to do because for ADPKD patients, a lot of time the cysts are just aligning with each other. It's kind of hard to separate boundaries. So yeah, that's something we are working on right now.
Okay.
So if there are no additional questions, I think we will end here. Thank you to all of the presenters, all of the listeners who asked excellent questions. And just to remind you to register for the PKD FASEB meeting. Okay, everybody,
Thank you.
have a great rest of your day.